In this video, we will look at sections 2.7 and 2.8, which also are on the molecules of cells. And in this particular case, we will be looking at proteins. So proteins are composed of monomers of amino acids, and each amino acid, it contains a central carbon atom, which is bonded to 4 different things. So each carbon atom is bonded to an amino group, which is the NH3+ that you can see on the left side of the diagram of each one of these. You also have a bond to a carboxyl group, which is a carbon attached to 2 oxygens, one of which is a double bonded oxygen plus one other oxygen. We also have a hydrogen bonded to each one of those carbon atoms. And so the way that one amino acid differs from another one is purely by the R group, which is on the bottom, and it's highlighted in kind of a bluish type of color there for you. So based upon the R group of each one of those, that determines the type of amino acid that it that forms. Otherwise, they are completely identical. So they have an identical structure of those 3 bonds, but the 4th one to the R group, those are different for each amino acid. So proteins, they perform numerous functions. For example, they transport molecules across a cell membrane. And one category of proteins called enzymes, they catalyze cell reactions. So they enable the reactions to take place in the cell, and they act as a catalyst in those particular reactions. Also, they provide structural support, for example, keratin in nails and collagen in tendons and ligaments. And so these materials are actually a protein. Then other proteins, they act as cell messengers, influencing metabolic activity in the cell. So peptides then, polypeptides are polymers of amino acids, which are joined by peptide bonds. And you can see there's an illustration there of the peptide bond. And peptide bonds, they are polar covalent bonds, and allows hydrogen bonding between amino acids. So this influences the 3-dimensional shape of the proteins. Then we have levels of organization of the protein, and the first level of organization is referred to as the primary structure, and this is just simply the amino acid sequence in the polypeptide. So, depending on the arrangement of the amino acids, that will determine its primary structure. Then we have a secondary structure, and this would be the polypeptide orientation, such as an alpha helix or a beta pleated sheet, which we will refer to again later. Then the tertiary structure, this would be the 3-dimensional shape of a polypeptide chain, and this shape is held together by covalent, ionic, and hydrogen bonds between the R groups on different amino acids. So, for example, a disulfide linkage between sulfur atoms between 2 cysteine amino acids would be one example of that. Then finally, we have the quaternary structure, and this is the 3-dimensional arrangement of polypeptide chains for proteins that have 2 or more chains. So with polypeptides with several chains to them, the way that these are arranged in their 3-dimensional structure, that is referred to as the quaternary structure. So the final shape of a protein is very important for its function, and proteins that become denatured, they undergo an irreversible change in their shape, and this causes them to no longer be active or to only be partially active, which means that they cannot carry out the function that they need to within the cell. So here's an illustration of the different structures. So the primary structure, once again, is just simply the sequence of amino acids, and then the secondary structure is due to hydrogen bonding between the amino acids, and allows it to form like an alpha helix in this case, or a beta pleated sheet. Then the tertiary structure, this is due in part to covalent bonding between the R groups. So giving the polypeptide twists and folds, etc., which gives it its characteristic globular shape. Then for those with several strands, then the way that these are arranged in a three-dimensional kind of form, that refers to the quaternary structure. All right, so again, here's a summary of what we've just talked about. And so we're now going to look at the nucleic acids, and there are two types. We have DNA, which everybody has heard about, and RNA as well. But so DNA, it stands for deoxyribonucleic acid, and this carries the genetic information of an organism. Then we also have RNA, which is the ribonucleic acid, and this consists of three different types actually. We have mRNA, tRNA, and rRNA. We will refer to them further, and we will define what each ones of those are. So the structure then of DNA and RNA, they are polymers of nucleotides, which contain a phosphate group, a pentose sugar, and a nitrogen-containing base. And so you can see in the diagram there, we have each of those. We have the nucleotide on the left in that diagram, and then we have a pentose sugar, and then the nitrogen-containing base. So nucleotides in DNA, they contain sugar deoxyribose and 4 different types of bases. And so we see only adenine, thymine, guanine, and cytosine. But the order in which we see them is what gives them their characteristic function. Then the nucleotides in RNA, they contain the ribose sugar and the same base as the DNA, except instead of thymine, now in RNA we have uracil. So nucleotides, they form a linear molecule called a strand, and a strand has a backbone of alternating sugar and phosphate groups with bases projecting to one side. So DNA is double-stranded with the strands twisted about each other in what's referred to as a double helix. And the strands are held together by hydrogen bonding. And the hydrogen bonding will always occur from cytosine to guanine and thymine to adenine. And RNA, however, is just single-stranded. Hope I said that right for cytosine. Might not have said that one correctly. The C represents cytosine. I might not have said that one correctly. So we have cytosine hydrogen bonded with guanine, and we have adenine, which is bonded to thymine, and once again, these are hydrogen bonds that bond them together. So again, here's a summary then of DNA structure compared with RNA. And so with DNA, we have a deoxyribose sugar. RNA, we have a ribose sugar. And then the bases in DNA are adenine, guanine, thymine, and cytosine. And then in RNA, we have uracil that replaces cytosine. DNA is a double strand, and RNA is a single strand. And so DNA forms a double helix, and RNA does not form a helix kind of a shape. So then we will look at briefly here at ATP, and we will be looking at this further in the course later on as well. But suffice to say right now that ATP, it stands for adenosine triphosphate, and it is this molecule that allows for the cell to obtain the energy that it requires to perform all the functions that a cell needs to perform. So it's a source of energy for chemical work, for the synthesis of macromolecules, for transport, for example, to pump substances across the plasma membrane, and also for mechanical work, supplies energy for processes such as muscle contraction and the movement of flagella within single-cell organisms. So the ATP then, it's involved in what's referred to as the ATP cycle, and this is how all cells derive their energy. And basically what happens is that energy is released when the bonds between phosphate groups are broken, and usually the last phosphate is cleaved to form ADP plus the phosphate group, and energy is used energy is then made available for reactions. So ATP then can be remade from ADP and a phosphate, which then allows for further energy to be used by the cell. So this is just very briefly on the ATP cycle, and we will be looking at that further as well later in the course. So here's a summary of what we looked at in this particular section. And so that concludes the main groups of the biomolecules, which include the carbohydrates, lipids, and proteins.